Welcome, curious minds, to the deep dive. Today, we're really embarking on a journey. We want to uncover one of life's quietest yet most profound miracles. How plants actually begin and grow. Think of it maybe as gaining X-ray vision into your garden, revealing the hidden machinery and sort of the ancient wisdom packed into every leaf and seed. Our source material for this fascinating journey is the Cells and Seeds, Basics and Beginnings chapter. It's from Brian Capon's Botany for Gardeners. So we're going to try and unpack the fundamental building blocks, the initial processes of plant life, and connect those complex botanical principles right back to what you see every day.
Yeah, and our understanding here, it actually goes back surprisingly far. You have to picture Robert Hooke, an English physicist back in 1665.
Wow, that far back.
Right. He's looking through, a pretty crude microscope for the time, and he looks at a slice of cork. He probably expects, just some uniform material, but instead he finds these astonishing, tiny, structured units, like little empty rooms, and he calls them cells, from the Latin cella, which means small room. It was completely revolutionary for the time.
I bet.
And Hooke's observation, it really laid the groundwork for what we now call the cell theory. This, profound idea that all living things are composed of one or more cells.
Okay.
It was as big a deal scientifically as the discovery of DNA was, much later.
Right.
This is really where the magic of understanding life at that microscopic level truly kicks off. So to really get a handle on what a plant cell is, try thinking of it like a super efficient factory, totally autonomous.
Factory. Okay, I can picture that.
Right. But not just any factory.
Yeah.
This one makes thousands of complex products from, really simple raw material.
Like what kind of material?
Just water, air, and soil minerals. Simple inputs.
Okay.
And its energy source, sunlight. Plus, if it needs to ramp up production, it could build an exact copy of itself, the whole factory, in just a day or two.
That's incredible speed.
And here's the kicker. Mentally squeeze that entire fully functioning factory into a tiny, tiny box, like 1/2000th of an inch on each side. About 0.05 millimeters.
No way. That's small.
That's your typical plant cell.
It's kind of mind-boggling. So much complexity in that tiny space. How does it manage all that?
Well, that's where the living machinery comes in. We call it protoplasm.
Protoplasm, okay.
And that includes the nucleus. Think of that as the main office or the control center. It sends out instructions for everything, operations, maintenance, reproduction.
Right.
And that's where the inherited chromosomes and DNA, the blueprints for new cells, are kept safe.
Okay, the blueprints, and then there's the cytoplasm.
Exactly. That's the sort of dynamic, jelly-like stuff inside where most of the cell's actual work gets done. It's bustling in there, and it's all held in by the cytoplasmic membrane. That's like a selective gatekeeper.
Selective.
Yeah, made of protein and fatty stuff. It controls what gets in and out. Water, food, minerals. Only the right things pass.
Got it.
And floating around in that cytoplasm like tiny machines are specialized organelles. Some are common to plant and animal cells, but plants have something special. Chloroplasts.
Ah, the green parts?
Exactly. These are the sites of photosynthesis,
Right.
where light energy gets used to make food. The green pigment, chlorophyll, is right inside them. So when you see a green leaf, you're really seeing the combined effect of millions of these tiny green bodies all busy capturing sunlight. It really makes that factory analogy come alive, doesn't it?
It really does. And there are other organelles too, right?
Oh, yes. You've got mitochondria, the powerhouses. They extract energy from food through respiration.
Okay.
And ribosomes, which are basically protein-making specialists, little workhorses.
Keeping everything running.
Absolutely. And then there's often a huge central vacuole.
A vacuole.
Yeah, think of it as a big membrane-bound sac, kind of a storage tank and maybe a waste dump.
What does it store?
Mostly water. It can take up like 95% of the cell's volume in a mature plant.
Wow.
And it also holds extra nutrients or tucks away toxic waste products safely.
And these cells, they don't just work alone, do they? You mentioned communication.
That's right. They have these tiny interconnecting strands of cytoplasm called plasmodesmata.
Plasmodesmata, okay.
They act like little bridges between cells, letting them exchange food and materials easily.
So they can coordinate.
Exactly. It makes the whole plant work together as one unit. Pretty sophisticated stuff.
Definitely. Now, outside of all that living protoplasm, there's the cell wall, right?
Yes, and that's crucial. It's rigid. It surrounds the protoplasm, protects it, and gives structural support to the whole plant.
Is that why plants wilt when the walls lose pressure?
That's often a big part of it, yeah. The walls rely on water pressure, turgor, for rigidity.
And here's a neat detail I read between the walls. Pectin.
You got it. The exact same stuff used to thicken jams and jellies.
No kidding.
It acts like a glue or mortar, binding the cells together. And the thickness of that wall, well, that determines how stiff that part of the plant is.
So a leaf has thin walls, but a tree trunk.
Exactly. Incredibly thick walls, especially with lignin added for extra strength. See, when a cell first forms, it has a thin primary wall, mostly cellulose.
Okay.
But as it gets older, it can add more layers inside that primary wall, more cellulose, and often lignin, this hardening stuff. That forms the secondary wall.
Lignin, that sounds familiar, like a lignified wood.
Precisely. Hardwoods like oak and ash, they owe their strength to tons of lignin in those secondary walls. But this leads to something really, really surprising about plant growth.
Oh, what's that?
As these walls get thicker and thicker, the space inside for the living protoplasm actually shrinks. It gets less access to water, to oxygen, and eventually, protoplasm dies.
It dies.
Yeah. It's almost like an act of self-sacrifice for the greater good of the plant structure.
Wow.
But here's the amazing part. Those hollow, dead cell walls, they keep doing their job. They provide support for the rest of the plant's life.
So wait, how much of a tree is actually dead cells?
Get this, up to 98% of a living tree's trunk and branches can be composed of dead cells.
98%. That's astonishing.
It really is. They gave their life, literally, for the structure of the whole organism.
Okay, so if cells are building these walls, and we see plants growing mostly upwards or downwards, how does the cell control its growth direction?
Great question. It's down to how those cellulose microfibrils are arranged in the walls.
The fibers.
Yeah. Imagine the cell as like an elongated box. On the long side walls, the fibrils are kind of coiled, running parallel around the cell.
Okay.
But on the end walls, they form more of a crisscross pattern.
Right.
So when the cell takes in water and starts to expand, the side walls can soften and stretch apart, letting the cell get longer.
But the ends.
The interwoven fibers on the ends resist stretching sideways, so the cell can't really get much wider.
So it mostly grows in length. That makes sense for stems and roots growing vertically.
Exactly. Which brings us to the two main processes of growth at the cellular level.
Okay.
First, you have cell division. New cells being made from existing ones.
Splitting in two.
Right. And critically, each new cell gets a complete identical set of genes through mitosis, that dance of the chromosomes.
Mitosis. Got it. And the second process?
That's cell elongation, what we just talked about, that period after division when the cell expands, mostly in length.
Okay, division and elongation. Where does this happen in the plant? Is it everywhere?
Not really everywhere all the time. Happens in specific zones called meristems. Literally means divided places.
Meristem.
You find apical meristems right at the tips, the apices of stems and roots.
The very ends.
Yep, and they're responsible for what we call primary growth.
Primary growth.
That's length. That's right. Increasing length. It's what lets leaves shoot up towards the sun and roots push down into the soil.
Like when you see a seedling just bursting upward.
That's exactly. Or a vine climbing. That's primary growth driven by those apical meristems. And it keeps going as long as those stems and roots are getting longer.
Okay. But plants also get thicker, right? Like tree trunks.
Good point. That's where lateral meristems come in.
Lateral side.
Yep. They run along the length of stems and roots, kind of like cylinders, and they're responsible for secondary growth, which is the thickening. Exactly. Adding girth, providing stability. Think about a tree trunk getting wider year after year. That's the slow, steady work of lateral meristems.
So you have primary growth for length, secondary for width.
And both types work together in a coordinated way to give a plant its overall shape and size. Though, day-to-day changes are tiny.
Yeah, you don't really notice it until years later.
Right. When that little sapling is suddenly a huge tree, it's quite the transformation.
It really is.
And sometimes you might see weird localized growth like galls.
Oh, yeah, those lumps on leaves or stems sometimes.
Exactly. That's usually caused by an insect or fungus stimulating the plant cells to divide rapidly right there. It's abnormal, but usually doesn't hurt the whole plant.
Interesting. Okay, so we've got cells, we've got growth. But all of this starts somewhere?
Right.
From the seed, right?
The seed, yes. This tiny package that seems so dry and lifeless.
Yeah, they don't look like much.
But that's totally deceptive. They're not lifeless at all. They're just dormant.
Dormant, like sleeping.
Sort of. More like suspended animation.
Yeah.
There's still measurable metabolic activity going on, just very, very slowly. They're waiting. And honestly, seeds are just nature's masterpiece of survival.
How so?
Well, they're compact, easy to store, incredibly tough. They can survive freezing drought conditions that would kill the parent plant easily.
Right.
Plus, their often drab colors help hide them from animals that might eat them. They're amazing little survival pods.
And the outer skin, the seed coat.
Yeah, that varies a lot. It can be thin or super thick and hard.
And that matters for germination.
Absolutely. It controls how fast water can get in, and water is key to waking the seed up.
So if it's too thick or hard?
Then it needs to be scarified, basically etched or scratched.
Scarified. Okay. How does that happen?
Well, in nature, it might happen slowly as fungi or bacteria in the soil break down the coat, or maybe faster if soil particles grind against it during heavy rain.
For animals. You mentioned that earlier.
Right. Seeds passing through an animal's digestive tract often get scarified. It's a win-win. The animal gets food, the plant gets its seeds dispersed, sometimes with a bit of natural fertilizer included.
Huh. Right. So gardeners sometimes need to do this deliberately.
Oh, definitely. For some seeds, you might have to nick each one with a file.
Tedious.
Can be. Or for larger batches, maybe shake them vigorously in a jar lined with sandpaper until coats are scratched up.
Like chipping away at a nutshell.
Pretty much, just giving it that little nudge to let the water in.
Okay, so let's go inside the seed. What's in there? You mentioned the embryo.
Right. Let's take a bean seed as an example. Soak it. Gently pry it open.
Okay.
You'll see two big fleshy halves. Those are the cotyledons or seed leaves, mostly food storage.
Got it. The fleshy bits.
But nestled between them, that's the main event. Tiny embryo. It's a perfect miniature plant.
Really? All the parts are there?
Yep. It's got a little root tip called a radicle, a short stem, and even a pair of tiny folded leaves. It's all curled up, ready to go.
Wow. A little plant in waiting.
Exactly. And during germination, that embryo is what grows.
Yeah.
Its apical meristems, root tip, shoot tip kick into gear, start that primary growth.
And the cotyledons, what happens to them?
They fuel the initial growth. They shrink as their stored food gets used up by the growing seedling.
Ah, okay.
And you can actually see different strategies here. With beans, the stem below the cotyledons elongates and pulls the cotyledons right up out of the ground. They eventually shrivel and fall off. That's epigous germination.
Epigous above ground.
Right. But with peas, for example, the cotyledons stay underground while the shoot grows up.
Yeah.
That's hypogous germination.
Hypogous, below ground.
Interesting. And that number of cotyledons, one or two, is actually a major way botanists classify flowering plants.
Oh, yeah.
Into dicots, meaning two cotyledons, like beans, roses, oak trees. And monocots, one cotyledon, like grasses, corn, lilies, orchids. Monocots are generally considered a bit more recent, evolutionarily speaking.
Huh. Who knew cotyledons were so important?
They are. And food storage isn't always just in cotyledons. Think of a corn kernel.
Yeah.
That's technically a seed inside a thin fruit wall. It has one cotyledon, but most of the food is in a structure called the endosperm.
Endosperm. Is that the soft white stuff in sweet corn?
That's exactly it. Or a coconut. The white meat is seed material, and the juice is liquid endosperm.
Wow, okay. So for planting these seeds, any tips?
Well, a good general rule of thumb for gardeners is plant a seed no deeper than its own length.
Its own length. Okay. Why is that?
Especially for small seeds, if you plant them too deep, the little seedling might use up all its stored food reserves just trying to reach the surface and the light. Going a bit shallow is usually safer than going too deep.
Makes sense. So what does a seed actually need to start growing? To germinate?
3 key things from the environment are crucial. Ample water, the right temperature, not too hot, not too cold, and well-aerated soil. It needs oxygen.
Oxygen? Why oxygen?
We'll get to that.
Yeah.
But first, remember, a dormant seed is incredibly dry, like less than 2% water inside.
Right, you said that protects it from freezing.
Exactly. So the first step is water uptake. It happens through a process called imbibition.
Imbibition. Sounds like drinking.
Kinda. Think of a dry sponge soaking up water. The dry cellulose and proteins in the seed just pull water molecules in.
Okay.
And as they absorb water, they swell up. This swelling is powerful enough to actually split the seed coat open.
Ah, so that helps get even more water and oxygen in.
Precisely. And once water's inside, the chemistry really starts humming.
What happens then?
Those large stored food molecules, starches, proteins, fats, get broken down by enzymes into smaller, usable units.
Like sugars.
Sugars, amino acids, yeah. Things the embryo can easily transport and use.
Used for what?
2 main things. Building materials for new cells in its growing meristems, and energy to fuel that growth.
Energy.
Yeah,
Okay, and that's where the oxygen comes in.
you got it. The embryo releases the energy stored in those food molecules through cellular respiration.
Just like animals do.
The very same process.
Right.
And it absolutely requires oxygen. That's why waterlogged soil is bad for germination. No oxygen.
Got it. So water comes in, food breaks down, energy gets released. What happens next?
Well, fueled by that energy, the embryo's root to the radical is usually the first part to emerge.
Pushes down first.
Yeah. It anchors the seedling, starts absorbing minerals, and importantly, takes up even more water through osmosis. We can dive into that another time.
Okay. Root first,
Then the shoot.
then the shoot emerges. Often it comes up with a little protective hook shape.
Oh yeah, I've seen that. Bent over.
Exactly. That protects the delicate young leaves as they get pulled up through the abrasive soil.
Clever.
And here's a really critical moment. A big switch.
What's that?
Up until now, the seedling has been heterotrophic,
Heterotrophic, meaning.
meaning it depends on an external food source, the stored food in the seed,
Yeah.
just like animals or fungi.
Dependent.
But when those first leaves unfold and reach the sunlight, bam, the switch happens.
To what?
It becomes autotrophic.
Oh, self-feeding.
Exactly. It starts photosynthesis, making its own food using sunlight. And from that moment on, for the rest of its life, it will produce its own food. It's a huge transition.
Wow. From dependent to self-sufficient. That's cool.
It really is. But germination isn't always that straightforward. Some seeds have extra requirements.
Oh, like what?
Well, some need a period of after ripening. The seed looks mature, but the embryo inside needs a bit more time to develop before it can sprout.
So it has to wait even after it leaves the parent plant.
Sometimes, yes. And this often leads to what's called staggered germination.
Staggered, like not all at once.
Right. Seeds from the same plant might sprout over months or even years. It drives gardeners crazy sometimes.
I can imagine.
But it's a brilliant survival strategy for the plant. If there's a sudden late frost or a drought, it doesn't wipe out all the seedlings at once. There's always a backup supply in the soil. Nature's insurance policy. Clever. What else can hold things up?
Sometimes there are chemical inhibitors right in the seed coat.
Chemicals that stop it growing.
Yeah. They need to be washed away, usually by heavy rainfall.
So the seed knows there's enough water for the long haul, not just a quick shower.
Exactly. It coordinates germination with really favorable conditions. That's also why you should always wash seeds you collect from fleshy fruits thoroughly. Get those inhibitors off.
Good tip. Okay, what else?
Then there's allelopathy. This is fascinating.
Allelopathy.
It's where one plant releases chemicals that actually stop other plants from growing nearby, sometimes even its own seedlings.
Really? Why would it do that?
Cuts down on competition for water, light, nutrients. Think about trying to grow stuff near a black walnut tree. Notoriously difficult.
I've heard of that. That's allelopathy.
That's a classic example. Those chemicals can come from falling leaves, twigs, even directly from the roots. It has potential for developing natural herbicides, too.
Interesting. Okay, more requirements.
Yep. Many plants from places with cold winters need stratification.
Stratification.
Their seeds need a period of cold, moist conditions before they'll germinate properly in the spring.
So they need to go through winter, essentially.
Basically, yes. Gardeners mimic this by putting seeds in a moist paper towel in the fridge for a few weeks or months. Artificial winter.
Right, stratification. Got it. Anything else?
Light sensitivity. Some seeds, especially from sun-loving plants, need exposure to red light to germinate.
Red light specifically. Why red?
While under a dense canopy of leaves, the light that filters through is low in red wavelengths.
Oh.
So, needing red light ensures the seed waits until there's an opening, maybe a tree falls or it's early spring before the trees leaf out, providing direct sunlight. It's a signal that conditions are right.
Nature's light switch. That's amazing.
Isn't it? And then you get into some really extreme requirements.
Like what?
Fire. Some plants, especially in fire-prone ecosystems like chaparral, their seeds actually need to be scarified by the heat of a wildfire.
Wow. Fire helps them sprout.
Yeah. The heat cracks the thick coats, plus the fire clears away competing plants and releases nutrients into the soil, creates perfect conditions for the seedlings.
Talk about resilience. Waiting for a fire.
It's an incredible adaptation. Or think about some desert wildflowers. Their seeds might need a pretreatment of heat, like being in hot desert soil, maybe 120 degrees Fahrenheit for a week or so.
To mimic the desert heat.
Exactly.
Yeah.
It ensures they germinate only during the cooler, rainier seasons when they have a better chance of survival.
So many specific tricks.
It really shows the diversity of strategies, and while gardeners often prefer plants that are easy to germinate,
Yeah, definitely.
understanding these unusual needs really deepens your appreciation for how plants have adapted, and it'll let you try growing maybe more unique, wilder species too.
That's a great point. So wrapping this all up, it's quite a journey, isn't it?
It really is.
From Hooke just glimpsing those first cells in cork, all the way to these incredibly complex, resilient seeds with all their special requirements. It's an amazing life story starting right there beneath the soil.
And understanding all this, it's not just abstract science, right?
Right.
It really does give you practical insights as a gardener.
How so?
Well, knowing about cell walls and growth patterns informs how and why you prune certain ways.
Okay.
Understanding dormancy and those special germination needs, that's huge for propagation, for starting seeds successfully.
Right. Knowing why stratification works or why some seeds need scarifying.
Exactly.
Yeah.
It guides your planting, your watering, even how you manage your soil.
Right.
It connects the why to the what to do.
It definitely deepens both the scientific understanding and the practical skills.
Absolutely.
So maybe a final thought for everyone listening. The next time you see a tiny seedling pushing its way through the soil, or you look up at a huge, sturdy tree, just take a second to consider those millions, billions of microscopic factories inside. Each one working away, building, adapting, reaching for light, pushing through soil. It's life's ingenuity on display.
It really is.
And it makes you wonder what other hidden biological processes are shaping the plants all around us, just waiting for us to look a little closer.
That's the exciting part. There's always more to learn.
So keep observing, keep exploring, and keep deepening that connection to the amazing plant world. Thanks for joining us on this deep dive. We'll catch you on the next one.